Welcome to Module 2. If Module 1 was about understanding the tools, the enzymes, now we focus on the workshops and production lines where these tools operate, the metabolic pathways. Metabolism is the sum total of all the chemical reactions in living organisms. It can be divided into two broad categories. Catabolism, breaking down molecules to release energy. Anabolism, building molecules, which requires energy. Think of metabolism as a city's economy. Catabolism is like the industries breaking down raw materials to produce fuel and parts. Anabolism is the construction sector, using those parts to build new structures. Enzymes are the managers and workers in this economy, directing traffic, ensuring each process happens at the right time, in the right place, and at the right speed. We'll explore three major categories of metabolism. Carbohydrate metabolism, protein metabolism, lipid metabolism. Carbohydrate metabolism. Carbohydrates are the primary source of quick energy in most organisms. The journey from a complex carbohydrate in your food to usable energy in your cells involves multiple well-orchestrated steps. Glycolysis, with structure. Glycolysis is the central pathway of glucose breakdown. It happens in the cytoplasm and does not require oxygen, making it an ancient and universal process. The word glycolysis means splitting of sugar. Here, one molecule of glucose, a 6-carbon sugar, is broken down into two molecules of pyruvate, each with 3 carbons. Along the way, a small but valuable amount of energy is captured in the form of ATP and NADH. We can divide glycolysis into two phases. Energy investment phase, the cell spends 2 ATP to prepare glucose for breakdown. This is like paying a small upfront cost to get a much bigger return later. Energy payoff phase, the cell produces 4 ATP and 2 NADH, ending with pyruvate. The net gain. 2 ATP and 2 NADH per glucose molecule, plus the pyruvate, which has its own future in metabolism. Fates of pyruvate. Once glycolysis is complete, pyruvate has several possible destinies, depending on the cell's oxygen availability and needs. 1. Lactic acid fermentation. In the absence of oxygen, for example, during intense exercise, pyruvate can be converted to lactic acid. This process regenerates NAD from NADH, allowing glycolysis to continue producing ATP anaerobically. The downside. Lactic acid accumulation can contribute to muscle fatigue. 2. Alcohol fermentation. Some organisms, like yeast, take pyruvate down a different path. They convert it into ethanol and carbon dioxide. Again, the point is to regenerate NAD so glycolysis can continue. This pathway is exploited in brewing and baking. The CO2 makes bread rise, and the ethanol makes beer. Pyruvate dehydrogenase reaction. In aerobic conditions, pyruvate enters the mitochondrion, where the pyruvate dehydrogenase complex converts it into acetyl-CoA. This step is like issuing a passport to pyruvate. It allows entry into the citric acid cycle. The reaction produces 1 molecule of NADH per pyruvate, 1 molecule of CO2 per pyruvate. Since glycolysis produces 2 pyruvates per glucose, these numbers double per glucose molecule. Citric acid cycle, with structure. Also known as the Krebs cycle or TCA cycle, this is a central hub of aerobic metabolism. It takes place in the mitochondrial matrix. Acetyl-CoA combines with oxaloacetate to form citrate, and through a series of enzyme-catalyzed reactions, the cycle releases 3 NADH, 1 FADH2, 1 GTP or ATP, 2 CO2. This is per acetyl-CoA, so per glucose molecule, 2 acetyl-CoA, the totals double. Beyond energy production, the citric acid cycle provides intermediates for biosynthesis, making it amphibolic, both catabolic and anabolic. Substrate level phosphorylation. ATP can be made in two main ways. Substrate level phosphorylation, where a phosphate group is directly transferred from a substrate to ADP. Oxidative phosphorylation, which we'll get to shortly. Substrate level phosphorylation happens in glycolysis and the citric acid cycle. It's a direct, immediate way to make ATP without needing the electron transport chain. Electron transport chain, ETC. Now we reach the mitochondrial inner membrane, the site of the ETC. Here, NADH and FADH2 donate their electrons to a series of protein complexes and mobile carriers. As electrons move through the chain, protons, H, are pumped into the intermembrane space, creating an electrochemical gradient, like water building up behind a dam. The final electron acceptor is oxygen, which combines with electrons and protons to form water. Oxidative phosphorylation. The proton gradient created by the ETC is harnessed by ATP synthase, a molecular turbine, to produce ATP. This process is called oxidative phosphorylation. For each NADH, about 2.5 ATP are produced, for each FADH2, about 1.5 ATP. This is where most of the cell's ATP comes from. Glycogen metabolism. Glycogenesis. When glucose is abundant, it is stored as glycogen, a branched polymer, in the liver and muscles. Glycogenesis is the process of synthesizing glycogen from glucose, using enzymes like glycogen synthase. Glycogenolysis. When energy is needed, glycogen is broken down into glucose 1-phosphate, which can be converted to glucose 6-phosphate for glycolysis. This process is catalyzed by enzymes such as glycogen phosphorylase. Protein metabolism. Proteins are usually not the body's first choice for energy, but they can be broken down into amino acids, which feed into metabolic pathways. Decarboxylation of amino acids. Some amino acids can lose a carboxyl group, COH, to form amines. This reaction is catalyzed by decarboxylases and often produces biologically active amines like histamine or serotonin. Deamination of amino acids. In deamination, the amino group is removed, usually producing ammonia and a keto acid. This is a key step in preparing amino acids for energy production. Transamination of amino acids. Here, the amino group from one amino acid is transferred to a keto acid, forming a new amino acid and a new keto acid. This is essential in amino acid interconversion. Urea cycle. Because ammonia is toxic, the body quickly converts it into urea for safe excretion via urine. The urea cycle occurs mainly in the liver and involves several enzymatic steps that capture nitrogen from amino acid breakdown. Glucogenic and ketogenic amino acids. Glucogenic amino acids are converted into intermediates that can form glucose via gluconeogenesis. Examples: alanine, glutamate. Ketogenic amino acids are converted into acetyl-CoA or acetoacetate, which can form ketone bodies. Examples: leucine, lysine. Lipid metabolism. Lipids are high-energy molecules, making them excellent long-term energy stores. Fatty acid biosynthesis, with structure. When energy is plentiful, the body synthesizes fatty acids from acetyl-CoA in the cytoplasm. The process requires NADPH and involves the fatty acid synthase complex. Oxidation of fatty acids. Fatty acid activation. Before fatty acids can be oxidized, they are activated in the cytoplasm by attachment to CoA, forming acyl-CoA. Carnitine shuttle. Long-chain fatty acids cannot cross the mitochondrial membrane directly, so they are transported via the carnitine shuttle system. Beta-oxidation of fatty acids. Inside the mitochondrial matrix, fatty acids are broken down 2 carbons at a time into acetyl-CoA, generating NADH and FADH2. For example, complete oxidation of palmitic acid yields a large amount of ATP due to its many acetyl-CoA units. Ketone bodies. When carbohydrate supply is low, as in fasting or uncontrolled diabetes, the liver converts excess acetyl-CoA into ketone bodies, acetoacetate, beta-hydroxybutyrate, acetone, which can serve as an alternative fuel for the brain and muscles. Cholesterol biosynthesis. The body can synthesize cholesterol from acetyl-CoA through a multi-step pathway in the cytoplasm and ER. Cholesterol is essential for cell membranes, steroid hormones, and bile acids. In this module, we've journeyed through the breakdown and storage of carbohydrates, the processing of proteins and safe nitrogen disposal, the mobilization and synthesis of lipids. Together with Module 1, you now have a complete understanding of how enzymes operate and how they coordinate the intricate network of metabolic pathways that sustain life.